the respiratory system, we learn about this trachea. This basically tracheal feature that you need to understand. This have got 4 layers, 4 coats. The hallmark of the trachea is that we have to identify the cartilage which is embedded in it within this particular layers of the trachea, which is a hyaline type of cartilage, looks like a smooth grass like or frosted glass like appearance. Other than this, trachea has this particular mucosa which is inner lining to it, and this particular inner mucosa have got a particular cell, we call them a pseudostratified ciliated columnar epithelium, and they are lining this particular mucosa of this particular trachea with 99 or 98% of the population. Apart from that, we have some other cells. We have these secretory cells, goblet cells, brush cells, etc. there. And then we have the submucosa which contains these mucosal glands. Now higher up in this respiratory system, we find this particular submucosa is fitted with this connective tissue like blood vessels, nerves, etc. along with the collagen fibers, elastin fibers, and these particular glands are plentifully seen. Now as we see this particular schematic picture and of this lung is that, see the trachea, you see that this particular submucosa is fitted by this particular glands which keeps on pouring the secretions within this particular lumens of the trachea or the upper respiratory tract. As we descend down from the trachea to the bronchus, bronchioles and respiratory bronchioles and alveoli, this particular productions of this particular mucus which is gradually settles down. So this particular element of this particular glandular element at the submucosa is gradually gets reduced or diminished. This particular space also gets diminished. This is replaced by this some kind of like a muscular element which is present, and collagen vascular element, cartilage we gradually lose down as we go down to this particular respiratory tract. So this particular thing that we are going to see, that evolutions of the respiratory system going higher up to down, this particular changes we are going to see. We call this as a bronchial tree. Our aim is to reach to this part of the lung which is having this kind of like a bulbous dilatation. We call them as alveoli. So we just like go through this particular airway tube, as you have seen that this is kind of like a bronchus which is splitting down into 2 bronchus. We call them as the principal bronchus, right and left bronchus, or for the right lung and the left lung we have the bronchus. Again for this particular lung, bronchus slits down into the lobes. The lungs have got like lobes, we have seen that, right lung, left lung, all of them have got lobes, upper lobe, middle lobe and this lower lobe and upper or lower. So they, according to this particular numbers of the lobe, this particular bronchus splits down, we call them as lobar bronchus. Then again it goes deep down and branch down into the segmental bronchus, which we have learned today in this particular region of the lectures, bronchopulmonary segment. So this particular segment we call them as bronchopulmonary segment. Each of this lung have got 10 such bronchopulmonary segment depending on the right lung and left lung which is we are studied. So this particular bronchopulmonary segment or segmental bronchi again branches down into terminal bronchioles, terminal bronchus, which is going down to lose this cartilage. As you see this, higher up to going down, this particular cartilage becomes scanty and just like a or becomes less or thinner, and slowly it's just like loses this particular texture as it goes down. So till this terminal bronchus we have this particular cartilage present. So this particular region up to this region from this particular trachea, they are having kind of like a more or less similar feature to identify as histologically. Till this cartilage is there, we can identify the respiratory trees or respiratory structure based on this cartilage is there. Once it loses this particular cartilage, then it has something else. This particular cartilage, it loses down beyond this particular terminal bronchus, we call them as bronchioles. And again bronchioles breaks down into the smaller branch, we call them as terminal bronchioles, which is again going to end up in alveoli, which is the dilated part of this lung parenchyma. Now as this particular element group breaks down into this bronchioles, here we have this particular muscular element becomes more, which is replacing this particular tracheal element or cartilage element. As you see, this cartilage is holding over there. Once this cartilage is abolished or evolutions of cartilage is happening there, lower down this particular region is more muscular compared to the upper end. Now being it more muscular muscles which is present in here, if you have any kind of constrictions of the muscles, it becomes spasmodic. That means this lumens remains in a collapsed state if it is constricting or contractions of the muscle happening. That's what happens here. if we have any kind of pathogens which is going to this end of this particular lung and creating some kind of trouble, this particular chemical substance, if it is produced, that irritates the muscles and make this muscle contract. If this muscle contracts, this particular part of this particular respiratory tree goes on spasm. That means it becomes narrowed down. So, lumen inside gets like a little bit constricted or super constricted. Then air getting in and air coming out becomes difficult. Then creates this particular problem or respiratory problem. We call them as bronchospasm. That means air getting in and coming out becomes difficult and creates a lot of sounds, a lot of things. And it is getting narrowed. The secretions which is coming from the upper ends gradually pours down and occupies this particular bronchioles to this particular alveoli and occupies this particular things and flooded this particular element. So, this particular air exchange becomes difficult. So, that is what happens here in case of this particular pulmonary diseases that we are going to learn in pathology, like asthma or COPD or any kind of like a lung disease which is terminally affecting this particular lungs, which is because of this particular high muscles which is present in this particular terminal bronchioles beyond that part, because of having more muscles. So, our aim of this particular class is to make a cross sections over here in this particular region and see microscope how this particular things looks like. So, what we are going to see in this particular cross section picture is that we are going to see this particular bronchioles which is devoid of this cartilages. So, we are going to see this particular circular element which is cartilage or round element or terminal or respiratory bronchioles which is there, which is ending into this particular sackless dilatations which is known as alveolar sac. So, if you cut this alveolar sac, we are going to see this cavity within this alveolar sac, and within this cavity of the alveolar sac, this particular respiratory bronchioles, small branches are ending into it. Other than this, we are going to see the blood vessels which are coming in this particular region for these gaseous exchanges. So, these three elements we are going to see. So, whenever you are microscopically seeing this particular part, we are going to see this particular lung parenchyma at this particular alveolar sac, how does these things look like. Then we are going to see this particular respiratory bronchioles, they are kind of circular elements which is having this kind of muscles present in it, but devoid of the cartilage which is present in it, and blood vessels which is present into it. That is what we are going to see. Now, histologically, this particular element, whenever you are seeing this particular pictures over there, we are going to see this particular pictures like this. So, this is a picture of the lung parenchyma which you are going to see or study today at the level of the alveolus, like lower down, bottom down, we are going to see this. So, this particular region as you are seeing here scattered over here is the alveolar sac which is split open, cut open. So, this is a alveolar sac which you are seeing here, another one, you are seeing from above this particular same bunch of grapes like appearance over here cut here, and seeing from top you are going to see this particular lung parenchyma like this. Into this particular lung parenchyma, you see that these are the circular element which is lined by this particular mucosal element, and then we have the submucosa which is replaced by this like a muscular element which is present in here. Cartilage is absent in this particular region which is also like muscular, and then we have the adventitia which is lying outside to it. Now, this particular element we call them as respiratory bronchioles. This is another, this is how it is there. And you see that there are inner lining which is present in here, which is which you can see here. The inner lining of this particular region which you are seeing, this inner mucosa of here, is replaced over here simple cuboidal epithelium cells. Higher up we see that the cells are tall, we call them as columnar cells. Here you see the flat tube like cells. আমি করলাম না, এই চলে গেল, কারেন্ট চলে গেছে, okay fine. So, that particular region we see that the cellular changes happens over here in the respiratory bronchioles, and this particular alveoli, as you are seeing this particular region, the lung alveoli is also visible in this particular into this particular region as well. So, what we are going to see here is terminal bronchioles that we are going to identify. We are going to see some blood vessels that we are going to see, and then we are going to see this alveolar sac. So, these are the three things that you are going to see and identify in the histological picture. So, whenever you are going to identify this particular tissues as lung tissues. These are the three things that you are going to see, and these are your identifying feature. That's like lung, we have talked, trachea we have talked about, this tube which has got three layers like this. So this particular terminal bronchioles, which is having this particular inner mucosa, which is lined by these cuboidal kind of cells, and no cartilage which is present in it. So one point about this particular terminal bronchiole that you are going to see is this: blood vessels which are going to see this capillaries, which is having a wide lumen, capillary with wide lumen. That means the cavity within this particular lumen is like this. How we are going to differentiate between these terminal bronchioles with this is that this particular thing is looking like this, like the circular tube or oval tube like this, they are looking like this particular blood vessels. In case of this particular terminal bronchioles, we have this particular layers which is present, which is thicker, and thick muscles are there present around it. So which is quite thicker compared to this one, and they have the muscles in it, and these particular muscles are responsible for contractions if it is irritated. So this particular lumen of these respiratory bronchioles and these particular blood vessels is differentiated, can be differentiated very easily by looking at the calibers and presence of this muscle layer which is thick in this particular respiratory bronchioles. Now coming to this alveolar sac, this particular alveolar sac as is looking like this particular element is bunch of grapes into which this terminal bronchioles are ending. Now if you cut this particular thing, it is thin walled sections we are seeing here. So this particular regions, we see that from the top is just like this, they are present like this. So this particular inner to this particular parenchyma of this particular region of alveoli present like this. It has a cavity or lumen like this inside to this particular roundiness, and they have a lining cells which is present in here. This lining cells which is present in here, we call them as pneumocytes. So this particular cells which is present in here holding this particular area, we call them as pneumocytes. Within this particular lining epithelium, we have gaps, we call them as angle of cone. So this particular region has a little bit gap which is present in here to communicate with this adjacent alveoli. That means air exchange or air can be traveling from A alveoli to B alveoli, so that they can be passing through the whole bunch of things. Now this particular pneumocyte which is basically covering this, they are resting on a basement membrane. So we have a fenestrate or a basement membrane which is present around it or sitting on it. They are not just like a flying, or they are not just like a hanging on air. So they are resting on this particular region, we call them as basement membrane, we call them as basement membrane over here is lamina propria. So this particular thing of alveoli have a thin wall basement membrane, we call them as lamina propria, which is onto which this particular flat cells are seeing here. This particular flat cells is looking like this. They are known as squamous kind of cells. So squamous type of epithelial looking like cells, they are present, and they are present in a series, and this particular cells which is present within this particular pulmonary alveoli, we call them as pneumocytes. Now there are two types of pneumocytes are there, pneumocyte 1 and pneumocyte 2. Out of this particular population of this particular pneumocyte, this pneumocyte 1 is likely like a 95 to 99%, and type 2 is only 1%. So this particular pneumocyte 1, which is basically present all over this particular pulmonary alveolar lining regions, they are making this particular alveoli resting on this particular lamina propria. And other variety is that type 2 pneumocyte. So this particular type 2 pneumocyte yields this particular type 1 pneumocyte. From the type 2 pneumocytes, it produces the type 1 pneumocytes. So this particular type 2 is very scanty, very less, and they basically give rise to this particular type 1 pneumocyte. Type 1 pneumocyte is lining everything around and making this particular alveoli. Now, other functions of this type 2 pneumocyte is that they produce a chemical substance, we call them as surfactant. So, this surfactant is a very important chemical substance, phospholipid element, which is secreted by this type 2 pneumocyte, which are placed like interspaced in small regions, small cells, they are present in between, they are producing or secreting this particular surfactant. So, functions of this particular type 2 pneumocyte is to produce this type 1 and surfactant. These are the two elements. The functions of the surfactant that we are going to learn in physiology and biochemistry, the chemical nature of it, they are very important element. Now, out of this particular pneumocyte, type 1 pneumocyte, which is the predominant population in this alveoli, they are looking like flat squamous kind of epithelium. They are lining over here. Now, other element that we need to understand over here. So, this area we have this particular air which is coming from this particular airway tube ending in here and reaching to a point where they can be transported or in or out to the blood vessels. That means, at this particular point they are reaching over here, docking in this region, living in this particular point and ready to be transported to the blood vessels in or out. That means, if it is carbon dioxide, that will be getting in and should be lifted up and getting outside, and if it is oxygen, that reaches to this point and getting to the blood vessels, so that through the blood vessels it can be transported rest part of the body. So, at this particular point we may have this particular the blood vessel capillary lumens ending like this. So, this particular area where you see that the blood vessels reaching to a point and merging with this lamina propria of this particular alveoli, they are making a point where this particular exchange or gaseous exchange can be taking place. So, this particular region where this gaseous exchange present over here, we call them as blood gas barrier. So, this particular region where this particular blood vessels coming in contact with this particular alveoli and making an exchange port where this exchange of the oxygen and CO2 can be done. So, oxygen, if it is coming getting in to this particular blood vessel and eventually going out, and CO2 from this particular blood vessel will be pumped into with exchange of this particular oxygen into this particular capillary end from this particular region will be exhaled out. So, this is how this mechanism works. Now, this particular region, as you are seeing here, these pulmonary alveoli meeting with this particular region with these blood vessels, we call them as, this region is that blood gas barrier. That means, there are a point where this particular exchange can be taking place, and this exchange over here is taking place between the gas only. So, that the blood vessels which have got like RBC, WBC, platelets, they are not exchanged at this particular point. So, only the gaseous exchange taking place. So, that is creating a barrier for the gaseous exchange only, not with the other particles. Or else, if you have something else, for example, if the RBC instead of the gas will be coming into this alveoli, that will be a disastrous thing. So, like the blood vessels which is bringing this WBC, if this WBC or white blood cells is coming from this blood vessel to this particular alveoli, that wouldn't be a good thing to happen, right. So, at this particular point there is a transportations of gaseous exchange taking place, and that is creating a barrier. Only gas can be allowed, they have the permit to go in and get out. So, this particular phenomenon which is restricting the gaseous exchange, creating a barrier, we call them as blood gas barrier. Now, how this particular blood gas barrier is formed? Now, this particular blood gas barrier is formed by this. You see that we have a thin layer of this pulmonary pneumocyte, and we have a thin film of coating chemical substance which is known as surfactant, which is produced by this particular type 2 pneumocyte. So, this type 2 pneumocyte creates or liberates a kind of chemical substance which is looks like the soap. So, this particular thin plate of soap layer which is present inside to it, right. So, this particular element which is known as the surfactant element which is present, which is guarding over here, and then we have this particular like the pneumocytes, and then we have this particular basement membrane, we call them as lamina propria over here. So, this lamina propria of this particular alveoli fuses with the lamina propria or basement membrane of this capillary. So, this capillary basement membrane and this particular lamina propria are fused at this particular point, and then we have this particular epithelial cells within this. So, this blood gas barrier is formed by inside to outside is surfactant chemical, then we have this lamina propria with alveoli, then we have lamina propria with the blood vessel or capillaries, they fused, and endothelial cells. So, these element they creates this particular barrier. Through this barrier only gas or CO2 and oxygen can be transported between or exchanged between. The other element like RBC, WBC they cannot go. Now, in some diseases if happens, say for example we have a bacterial attack or viral attack, this blood based barrier which is created by these elements may be destroyed. That means this selectivity of these molecules which is going in here is no more there if they are destroyed. That liberates or that creates the freedom of this red blood cell, white blood cells, or these elements which is present in here, protein substance which is present in the blood can migrate from here and end up in the alveoli, and that creates the problem if it is getting in here. So, we have a loss of blood based barrier in some infections, say for example pneumonia, you have an infection, this blood based barrier is destroyed by these pathogens or diseases. Then we have the trouble like blood is getting inside and this patient is coughing out blood. So, this blood, how this blood is coming when you are coughing, it should be bringing some kind of mucus which you normally secrete. Instead of that you are getting this particular blood coming out through it. So, that particular blood is coming from breached blood gas barrier, the leaking of this particular element, red blood cells, white blood cells getting in mixed up with the mucus. When you are coughing out, the mucus along with this blood is coming out. Get these things how it is coming. So, this is how we have to understand light is there. So, this is how this blood gas barrier is important. So, it is important short note that you have to prepare for examination blood gas barrier with this schematic picture of it. In physiology they ask this question, in anatomy we ask this question. So, it is an must know region in this discussion whatever we are going to do today. About theoretical aspect of today's class is blood gas barrier, how this blood gas barrier matters to us. You will learn in physiology detail about it, they have a detailed chapter about. The Gannon book is very good on this particular region. Surfactant, another thing that you have to understand this particular aspects of this. So, this is how we are seeing here this particular cross sections of the lung parenchyma. We call them as parenchymal tissues. These are the alveolar sacs. They are the lining cells which is present in the alveolar sacs. That means pneumocytes. Pneumocytes resting on the basement membrane which cannot be seen, that needs higher magnification. Now, this particular element have got multiple air sac called alveolar sacs are there. They are communicated with each other by some pores, we call them as pore, Cohn's pore. And eventually this particular element as we are seeing here is this particular terminal bronchioles or respiratory bronchioles which are bringing this particular air, and this particular air getting into this cavity of the alveoli. Once they are inside, they can be exchanged with this gaseous exchange. This is another one respiratory bronchiole, this is another. You see the caliber, this is bigger, this is smaller, that means it is breaking down, branching down, etc., etc. So, this alveoli, alveolar cavity you should be able to identify. So, these cavities, these are alveolar cavities, and this is what we are seeing here. This is a wider lumen you are seeing here, that where you see this particular section is taken, where you are seeing this little bit in a higher level cross section which are taken out here. Now, out of this you should be able to identify this particular element as the respiratory bronchioles. You should have a lining cells. These lining cells, by theory you know that this is no more a column cells, these are cuboidal cells which is present, and the muscles which are present in here which is quite significantly enlarged or accentuated compared to this particular trachea that you have seen, which is predominantly have the trachea cartilage which is present in here. So, this particular respiratory bronchiole and along with this particular pulmonary alveoli bronchiole, these are things that you are going to see. When you are going to see this particular lung tissues in this particular cross sections, you are seeing this cross section picture like this. So, what you are going to see this particular thing is that bronchioles, they are of different sizes, medium, terminal, respiratory, they are the smooth muscle cells, they are present within this particular bronchioles, they have a lot of connective tissue, but do not have any cartilages which are present in here. and within here we see that this particular area we have the stripped open of this particular alveoli or cavity of the alveoli. They are communicated with each other, alveolar sacs between them. They have a pattern of airflow which is going through in this particular region between these alveolar cavities. So, what you have, this particular alveoli is made of this alveolar epithelium. They are of two types, type 1, type 2. You cannot forget about type 1, type 2 pneumocytes. Type 1 is the predominant one, type 2 is the less one. Type 2 produces the one and as well as produces surfactant. This one you cannot forget about. So, functions of the type 2 pneumocytes is very important that you have to remember in these regions. And this particular, other than this, we have to know other cells which are present as well, like they are present with this particular cells, we call them a basal cell, brush cell, Clara cells. They are defense kind of cells which is present in here other than this type 1, type 2 pneumocytes. So, non-alveolar cells, they are present in this particular respiratory tract. They have different types, goblet cells, mucosecreting cell, brush cells which is brushing this particular regions, etc., etc. So, these alveolar macrophages or defense cells which is present in here, they remove the dust particles if they reach to this particular alveolar this depth. So, normally we see that as it is travelling down to this particular respiratory tract, whenever above, from above below. So, when it is gradually reaching here, this usually this air particles which is having a particular nano diameter usually gets trapped through here. If eventually they reach to this particular lower part or bottom part to the alveoli, they have the special scavenging cells, we call them as alveolar defense cells. In physiology, we are going to learn that particular things in detail. So, the alveolar macrophage cells, they digest or grasp this particular food part, that particular dust particles, and this particular alveolar macrophage cells becomes dust cells. They trap this carbon tar, any pollen grains, etc., etc., into this particular region. So, this is about this particular thing, whatever we have talked about the histoanatomy. Now, we are talking about the blood air barrier. Normally, gaseous exchange is permitted, anything beyond is not permitted. So, that particular permissions is happening between this particular barrier which is present in here. So, this particular barrier is formed by these things. This is the lumen of alveoli that you are seeing here. This is the capillary which is nearby freezing here in this region. Now, you see that inside to it we have a thin layer of the surfactant, then we have this particular basement membrane, we call them as lamina propria. What happens over here, this lamina propria here and this particular basement membrane of this capillaries lumen, they fuse together, and then we have this endothelium lining here inside to it which is present within these capillaries. So, capillary endothelium with this basement membrane along with this pneumocytes and along with pneumocyte cells along with this thin layer of capillary, they together make this particular blood gas barrier. So, normally they allow this particular gas to permit or travel between the CO2 and oxygen exchange can be traveling happening between them. These red cells WBC, RBC cannot normally flow through, neither the protein particles or fat particles which are present in this particular lumens, they are going in. What happens if you have damaged this particular blood particle or this material which is present in these lumens of the capillary, they can get inside and create the problem. So, that we call them as loss of blood-brain barrier, then we have these troubles. So, what you have to know about here is that what this particular blood air barrier or blood gas barrier is consisting of, what is the normal function of that, and what happens if you have a loss of blood gas barrier. So, this element you must prepare it as short note. Go home and prepare this as a short note, write it down, read from books and make a gist of it and prepare it very well, because these are very important elements that you are going to learn. And other than this, we do not have much of theoretical discussion left about this particular lung tissue. So, whenever you are going to see the lung tissue, that thing which you have to focus here, if it is a lung tissue, you have to identify it as a lung tissue. What are the things that we are going to see? This label where you are going to see the lung tissue is that lower alveolar level. So, what you are going to see this is that alveolar cavity which is lined by the pneumocyte that you are going to see, and terminal respiratory bronchioles which are ending on it, devoid of the cartilage which is present in here. So, circular element that you are going to see, and along with that we can see the blood vessels if they are falling on that particular cross sections. So, these things you are going to see. Then this particular alveoli is lined by the cells which you know, type 1 predominantly and type 2 is less. Type 2 has a specific function to secrete a substance, we call them as surfactant, which is very intensely important substance for us. and along with that this particular respiratory bronchioles have got more muscular element than that of cartilaginous element, right. হ্যাঁ তো সেগুলো অ্যালভিউলার স্পেস কোথায় তোর এখানে অল अराउंड কি আছে অ্যালভিউলার স্পেস আর বুঝি যেমন করে কেমন করে আছে ছবিগুলো কোথায় কোনটা অ্যালভিউলার